Hey everyone, this is Saul, and I am presenting my research, which was investigating the biodegradation of synthetic dyes by Pseudomonas putida using spectrophotometric and chromatographic analysis. The two dyes that I used were Congo Red and Red 40, also known as Allura Red AC. Both are azo dyes and are used in food and textile, but once they're in the environment, they can break down into toxic aromatic amines. Congo Red is very well studied, but Red 40, even though it is used a lot in food, is not that well studied. So I'm using P. putida, which is found in soil and wastewater and has versatile metabolism and is resistant to a range of toxic dyes to study the breakdown of both of these dyes. Spectrophotometry was used to monitor the decolorization, and UPLC chromatography was used to identify the breakdown products. My hypothesis was that PP data would biodegrade Congo Red and Red 40 under the aerobic conditions, and aromatic intermediates would be detectable by UPLC. For the methods, the Putida bacteria was grown on nutrient agar and then transferred or inoculated into nutrient broth and then transferred into M9 media. The bacteria in the M9 media were then treated with different concentrations of both the dyes at 550, 150, and 300 milligram per liter, amongst which only the 150 milligram per liter was done in biological triplicates. MIC assay was also done to test the toxicity of the dye at different concentrations, and it was found that even the highest concentration of dye did not hinder the growth of the bacteria. The growth was measured by OD600, and decolorization was measured at the absorbance max for each of the dyes, which was 947 nanometers for Congo Red and 504 nanometers for Red 40. The samples were then analyzed by UPLC against a benzene standard. If you take a look at the growth data, we can see that pputa grew well in both the dyes, and neither dyes um, hindered the growth of the bacteria. This shows that the bacteria is very tolerant under the experimental conditions in this case, and this this agrees with a lot of the literature that says that P. putida is very tolerant. When it comes to decolorization, though, we have some interesting data. When it comes to the Congo Red, we can see a significant decrease in color as we move from the starting conditions to 96 hours. And once statistically analyzed using two-way ANOVA, the decolorization is shown to be statistically significant in both time and treatment interaction. When it comes to Allura Red, though, there is no change in the color. There's little to no decolorization, and once statistically analyzed, it is shown that it is not statistically different at all. When it comes to the UPLC, the lower concentrations of the benzene standard did not show clear peaks, but the 300 and the 500 microgram per ml standards show two very clear peaks at 4.5 and 5.5 minutes. However, the biodegradation samples taken, not a single one of those samples showed the same peak as the benzene standards. This shows that we cannot confirm any benzene related byproducts from the biodegradation. To conclude, P. Putida was able to grow in both the dyes, but only Congo Red was broken down and decolorized significantly. Allura Red showed little to no change, and the UPLC results did not confirm any benzene related products and needs to be utilized further. Um, if I were to keep or continue the study, I would 100% um, optimize the biodegradation conditions as well as the UPLC methods. I would test at different concentrations, different oxygen conditions, increase the replicates for every single concentration, as well as work a lot more on improving the UPLC methods so that um, a biodegradation product is measurable. These are the references, and lastly, I would like to acknowledge um, my thanks to my professors, which is Jessica and Mike. They have been very good at guiding me throughout the research, but I would also like to thank Marla, Crystal, and Shanda for being a great support during the entire research. Thank you.